Colorectal cancer is the most common cancer and the second most common cause of cancer-related death within the European Union in men and women combined. Hence, an increasing number of countries recommend and implement CRC screening. There are 3 screening methods: screening by fecal occult blood testing, flexible sigmoidoscopy, and endoscopic screening. The first 2 screening options will have to be supplemented with additional tests if results are positive. Colonoscopy needs no additional screening tests. In addition, polypoid precursor lesions in sporadic CRC can be removed during the procedure and thus improve survival. However, available estimates of uptake rates and mortality-reducing efficacy of all the 3 screening methods are similar, showing a 20 to 25% reduction in CRC mortality. Despite these results, colonoscopy is being increasingly used as the primary screening method in Europe and the US. Since colonoscopy is expensive, invasive, and inconvenient, its benefits need to be studied in a randomized trial mimicking organized population-based screening. Only such an approach would allow to quantify both benefits and adverse outcomes of colonoscopy screening and valid comparison with other screening modalities. The need for and design of such a trial are the issues addressed by Kaminski et al. In the recent paper in the July issue of Endoscopy entitled The NordICC Study, Rationale and the Design of a Randomized Trial on Colonoscopy Screening for Colorectal Cancer. Background and study aim. While colonoscopy screening is widely used in several European countries and the United States, there are no randomized trials to quantify its benefits. The Nordic European Initiative on Colorectal Cancer, NordICC, is a multinational, randomized, controlled trial aiming at investigating the effect of colonoscopy screening on colorectal cancer incidence and mortality. This paper describes the rationale and the design of the NordICC trial. Study design. Men and women aged 55 to 64 years are drawn from the population registries in the participating countries and randomly assigned to either once-only colonoscopy screening with removal of all detected lesions or no screening, standard of care in the trial regions. All individuals are followed for 15 years after inclusion using dedicated national registries. The primary endpoints of the trial are cumulative CRC-specific death and CRC incidence during 15 years of follow-up. Power analysis. We hypothesize a 50% CRC mortality reducing efficacy of the colonoscopy intervention and predict 50% compliance, yielding a 25% mortality reduction among those invited to screening. For 90% power and a two-sided alpha level of 0.05, using a 2-to-1 randomization, 45,600 individuals will be randomized to control and 22,800 individuals to the colonoscopy group. Interim analysis of the effects of colonoscopy on CRC incidence and mortality will be performed at 10-year follow-up. Conclusions. The aim of the Nordic trial is to quantify the effectiveness of population-based colonoscopy screening. This will allow development of evidence-based guidelines for CRC screening in the general population.